think we'll start. I unfortunately have some bad news. One of the speakers had a family issue and had to go back, so Maya won't be talking. We're just going to compress everything toward the front and finish a little early. I know all of you are disappointed because you don't want to be here until 5 o'clock, but that's the way it's going to work out. The other mixed blessing was that John Mazziota, the Vice Chancellor, was supposed to open our fall symposium, and he was called away unexpectedly. But because it's a mixed blessing is we have Marie-Francoise Chasselet, our former chair, who will say a few words. And I think this is a wonderful opportunity. She is the person who started these fall symposia. And I think it's become a really good and nice tradition that we have in the department. It's a welcome back for people after the summer, and it's a chance for us to learn about what each of us are doing in our laboratories. UCLA is a big place, as you all know, and people sometimes don't talk to each other for years. And the idea of getting together once a year and exchanging ideas is a fabulous, fabulous thing. And so I'm going to allow Marie-Francoise a few minutes to tell us about her thoughts behind the symposium. And what you should know is, after Marie-Francoise left here, she wanted to relax for a while. So to relax, she's going to be interim chair of neurology.
Yeah, it looks like, taking over from Mazziota is the story of my life. So I'm not surprised to be here and feeling for John, since many of you may know that 6 months ago I moved to the dark side and started chairing the Department of Neurology. So I'm very, very honored to open the symposium this afternoon. As Paul said, that was something we started in 2002, and we felt it was a great opportunity to get together and to hear about our own work, what happens really in the hallway next door. Because we often go and give talks all over the world, and we invite people from all over the world. It's a privilege we have to be in a great and big place like UCLA. And sometimes we don't know what people next door are doing. And I think this is really the theme of this symposium has always been to focus on the work done in the Department of Neurobiology. And I'm delighted that Otis and Paul Majsewicz have decided to continue this tradition. A very important part of the tradition is that there is supposed to be a really great party at the end. So remember that and don't go away, stay for the afternoon, there will be really good food. That's what you get from having an event started with a French chair. And so I don't want to take more of your time. I'm really delighted to see all of you here. And I think you are in for a treat and a really fascinating afternoon. And thank you, Paul, for the opportunity to open the afternoon. Have a great time.
Thank you, and unfortunately, before Dean gets a chance to speak, I want to make a couple of announcements. The first announcement is that our former colleague, Artie Scheibel, has been very generous and has presented the department with a very nice gift. And further details will be evolving over time. The other announcement, which we sent out from the office, was that Dean Bach was awarded the Helen Keller Prize in Vision Research. This is a very, very big award and richly deserved by Dean. And as has been the tradition now over the last couple of years, we have some prizes to give out. We have two awards that are aimed at students in the department who are doing research. One is the Charles Sawyer Memorial Travel pressure of the first class of medical students in 1951. And upon his passing, this memorial award was started, and we now, this year, give the award to Christopher Octo. So Christopher, are you here, please? Someplace? Can you come up here, please? I'd like to give you a certificate. Another award is the Lawrence Kruger Undergraduate Scholarship Award. And we all know Larry. I think I just saw him walk in someplace where he's right there in front of you. Couldn't be any more clear. So, Larry, thank you. There is a foundation that supports an undergraduate scholarship for people in neurobiology labs that are working, so that they have some money over the summer to enjoy what we get to do for a living our whole lives. And this year, the awardee is Shagoya Azar. All right, and now, the moment we've all been waiting for. The first talk of our symposium will be by Dean Buonomano. Dean is the vice chair of our department and also a member of the Department of Psychology. So, Dean, I'm going to let you now hold forward. And with time you stood.
Fair enough. Thank you for inviting me, Paul, and thank you, Marie Francoise, for starting this tradition. Since I've become vice chair of the department, I've become inordinately concerned with Paul's health. Have you been exercising? Okay, so by the way, I'm going to be giving a very similar talk at SFN, at a symposium, so if anybody has any advice for me, that would be very much appreciated. So as I think many of you know, can everybody hear me by the way? Okay. So as many of you know, my lab is interested in how the brain tells time. And time is one of those words that's very difficult to... So I paused my speech there for a moment on purpose to make 2 points. The first is that you're automatically telling time. You're automatically creating a temporal expectation of when the next word should come out of my mouth. When that expectation is violated, we end up with an awkward silence, in which you were probably thinking, uh-oh, Dean's having an absence here. The second point is that many of you probably completed the last word for me. Time is one of those words that's very difficult to define. And that's because I would put forth that one of your brain's main functions is to predict what's about to happen, to predict both the short term and long term. Because how well we predict what's going to happen in both the short term and the long term translates into the evolutionary terms, either survival and reproduction. So in order to predict the future, the brain needs a series of clocks. It needs to be able to tell time. And the brain has many ways to tell time. And we tell time over a vast scale of over 12 orders of magnitude, from microsecond processing to cell localization to circadian and infradian rhythms. But I'm going to focus here on what we call perceptual timing, timing that takes place on the scale of hundreds of milliseconds to a few seconds. See?
Is this working?
Okay. I'm just going to amplify the sound here coming from my computer. So the most trivial interval temporal passage used in the lab is an interval or duration discrimination. So for example, in music report.
Is this working?
Yeah.
Okay, so can you hear that? Yeah, 2 beats. So we can say that's like a 1/8 note in music versus a 1/16 note. And that's a very trivial duration discrimination path that everybody can understand and see that the second tone was the longest. So that's something that everybody should be able to perceive. But One of my favorite examples of how sophisticated the brain's ability to tell time is that we can reduce language, we can communicate, we can talk to each other through a purely temporal code. And that's what Morse code is. So I like this example in Morse code. And in Morse code is a temporal, you reduce language to a purely temporal code because you have to tell the dashes from the dots, that's a temporal task, but you also have the overall temporal structure of the stimulus. So I like this example because you can see that the sequence of symbols is exactly the same. Dash, 4 dots, 2 dashes. Dash, 4 dots, 2 dashes. The only difference between the dat and the dim is the pause, the so-called inter-letter interval. So to the untrained ear, including mine, that's very hard to make out. And if I play this, you can probably hear the inter-word interval, the break between that and dim. So let's see if we can hear that. So you can probably hear the pause between the words there, but the rest probably is sort of a continuum of bits and dashes that would be hard for you to pick out without practice. And indeed, that's what it takes. But the timing, so all that seems like to be a very challenging task. It's really no more challenging than what you're doing right now, which is understanding what you talk, which is speech. So an example I'll use here of speech is how the pause, the so-called phrasal boundaries in speech, you're processing automatically. So if I say, they gave her cat food, or they gave her cat food, there's two different meanings, there's two different semantic contents that you sort of pick up automatically in that situation. In speech, of course, or music, you can't even fathom what they would be without the way they tell time. So one of the things we're interested in is how does the brain accomplish these tasks, both simple interval discrimination and complex temporal tasks, both at the sensory and the motor side. Now, I'm going to discuss this, and here's the outline. I'm going to talk about what we call state-dependent network model planning, some experimental tests of this model, and then some more theoretical models of motor timing and neurodynamics in recurrent neural networks. So when we think about telling time, we're inevitably biased by the notion of man-made clocks, of clocks that function based on an oscillator, such as a quartz crystal or a pendulum, and another system that's just counting each one of those ticks. But in reality, any dynamical system that evolves in time can be used to tell time. And the brain, of course, is probably the most complex dynamical system in the known universe. So it's not surprising that it could use its dynamics to encode time. And here, an example of what I mean by that is a liquid. A liquid is a physical system. And because it evolves and changes in time, it naturally encodes time. So if I ask you which one of these raindrops fell first, you can easily tell me that that raindrop fell first. Because time, temporal information, is written in the state of the system. And with a bit of calculations, you can tell me the interval between those two raindrops, the inter-raindrop interval, if you will. So the state of the system is key. So temporal information is encoded in the state of the system. So the idea is that the brain encodes time in its state. But what do we mean by the state of a neural network? So normally, what people think of when we talk about the state of a neural network, the state of the brain, they think of which neurons are active. So we'll call that the active state. So here in my little diagram, I have excitatory neurons and inhibitory neurons, and which neurons are firing are shown in white. So the active state here is defined by the vector of 1, 0, 0, 0, 1, 1, 0, 1, 0, 1 of neurons that are firing. So here is a recording from the locus of 112 projection neurons changing in time. And the state at each one of these time bits is different. So in a sense, you can tell how much time has elapsed by which neurons are firing, assuming this is a reproducible repetitive pattern. So that's the active state. But then the brain is much more complicated to define the states just by which neurons are firing. Neurons and synapses have many time-dependent properties, such as short-term synaptic plasticity, slow network receptors, and channels of slow time constants. I refer to this as the hidden state of the system, hidden state of the network, because what's happening here is more or less invisible to the electrophysiologist measuring the vestigial electrodes. We don't have access to this information as well as we have access to this information. And my favorite example here is short-term semantic plasticity, shown here from the paper from Alex Reyes and Bert Sackman. This second ESP is bigger than that first ESP. So it contains information about the passage of time, doesn't it? The fact that it's bigger here or here reflects how much time has elapsed since the last action potential. And this time structure changes with that interval. So let me give you an example of how we could use the hidden state of the network to do an interval discrimination task. Suppose we have an interval discrimination task in which we need to discriminate a short interval versus a long interval, or a short note versus a long note. So simple interval iteration discrimination task. Now, let's look at the simplest circuit we can come up with. Just two neurons, an inhibitory and excitatory neuron, that both receive an input that conveys the input, that conveys a beep beep to the system. So because this synapse has short-term semantic plasticity, that ESP might be bigger than that one, and that one, and that one, and it TKs. So you can see that if we wanted to enforce our threshold for this neuron right here somewhere, that this neuron would become a 50 millisecond detector. So this is 50 millisecond stimulus, 100 millisecond stimulus, 200 millisecond stimulus. These traces are overlaid, okay? So we can make a 50 millisecond detector by putting our threshold in the right place. So that's what we're doing right here. So if you have set your threshold so that it's right above threshold at the second, the second ESP at 50 milliseconds, this neuron can tell you the interval of that stimuli. So this is fairly obvious. Slightly less obvious is the fact that you can create many different detectors with this circuit by playing with these weights appropriately. Now you can increase the weight onto the inhibitory neuron. And the inhibitory neuron now fires at 50, vetoing the would-be action potential here. And because you decrease your threshold further, now this neuron fires for 100 milliseconds. So even this trivial circuit here, with short-term plasticity, with hidden state, can be used to solve a long-standing problem. How we can do a simple interval iteration discrimination test. And by the way, if you have any questions during the talk, please feel free to interrupt me. So we've shown a while back now that now if you explore synapse space, you do a parametric analysis of the strength of all the synapses here, that one, that one, and that one. And I'm just going to show you the data here with the input to the excitatory and the input to the inhibitory. We have these transitions from neurons that respond to 50 millisecond, 100 millisecond, 200 milliseconds. So the color is coding the interval selectivity. So just by having this, what you're doing is you have a dynamic change in the balance of excitation and inhibition. So short-term semantic plasticity ensures that the balance of excitation and inhibition changes from the perspective of the postsynaptic cell, changing its temporal properties. You can't get that without changing both the balance of excitation and inhibition. Now, this is a very trivial circuit, and this is fairly, we proposed this a while back now, but more recently, there's now significant experimental evidence that in some systems, this is how organisms tell time. So this is an example from Bruce Carlson's lab who studies electric fish. Electric fish use temporal information, temporal pulses, to communicate with each other. So the timing of electrical pulses conveys information about social behavior. So here's a neuron in the brainstem that does not respond very well to 100 millisecond pulses, electric pulses 100 milliseconds apart. Responds fairly well to these action potentials here, 3 trials. A pulse is 50 milliseconds apart, but not 10 milliseconds apart. The evidence is that this is due to the balance, change in the balance of excitation and inhibition imposed by short-term semantic plasticity. And there's other studies now that support that notion. But those in very reduced circuits, the real goal of these state-dependent network models is to propose a general framework for how the brain performs computations. So now let's look at a large network. A network in which you don't need to go in and tune each weight individually. synaptic strength individually. So we go back here to our network with side-turning and return neurons, and it receives an input. So this is suppose our peak, the two tones are discriminating. This tone will come in and will activate a population of neurons. Over this point in time here, right before the second tone, let's assume that the active state of the network goes back to its rest state. So the active state is here, is the same here and here. So if you were just looking at the active state, you would think this network is in the same configuration. But we know, as I just told you about the hidden state, that there's a memory of what happened over here. Because some synapses are facilitated, some synapses, some neurons might have after hyperpolarizations and so forth. So there's a memory in the hidden state of what happened in the past. So when you have the next input coming in, the response of this network should be different from here, even though it's the same network, it's the same input, because the state was in a different state. Because the synapses are stronger, other synapses are weaker. So in simulations, we can show that when you take that into account, the response, this again, this is now looking at each one of these is a simulated unit in spiking units. That's the voltage in red. Red is like, dark red is hyperpolarized, red and white is depolarized, whites are spikes. So that's the response to the first pulse, first tone. That's the response to the second tone at 100 milliseconds, or the second tone at 200. And the question is this active state different from that active state, different from that active state? And we can show that it is. There's some neurons here that just respond to 100 milliseconds, others that respond preferentially to 200 milliseconds. And then we can create our output detectors that are specialized detectors for particular temporal intervals or patterns. So, what we advocate is that we need to think about the brain not as operating in independent time slices, but as a dynamical system in which the cortex encodes each stimulus in the context of the previous stimulus. So it doesn't make sense to look at the brain in response to stimulus A without asking what preceded it. And this, of course, is what you're doing in speech. Every phoneme I'm saying is interpreted not as an independent sound, but in the context of what preceded it, and that makes words. So this is a bit of a background here. Now, the prediction of what we call this state-dependent network model is that timing is a local and intrinsic computation of neural circuits. Now, to give you some context, in the timing field, there's two frameworks in which people have thought about how the brain tells time. So for decades, psychologists have advocated that, well, maybe you have some specialized part of your brain that's dedicated to timing. So here I'm drawing as a cerebellum, but it could be anywhere. But there's some specialized circuit or area that's responsible for time. Sort of like the chips on our computer, you have a clock chip that's responsible for keeping time. The opposing view is that time is such an important aspect of what the brain needs to do that virtually any circuit in the brain can tell time if it needs to. Yeah, Josh.
In the previous slide. Yeah, so you've got the initial state in the stimulus, and after the stimulus it returns back to what looks like the initial state, it's got the state states, it's not really stimulus. What happens if you just keep doing the same thing again and again and again? Yeah, so then, stimulus 3, it seems to do the same thing as the hidden state stimulus 2.
So how many times did you say again?
6.
So the point, I don't think it was
My question to you, how many times can I say that?
So my point, perhaps overly obscure, why I answer to Josh, was that the network will reach a stable state. But that's exactly what we do in behavior. If Josh says again and again and again 6 times, or again and again and again 7 times, it's hard for us to keep track of that. And the prediction is that it's hard to keep track of that is precisely because, yes, it changes state for 1 and for 2, but after a while it does converge to a steady state where you lose that information.
So you think the output of the network would be consistent from trial to trial to trial?
To trial, maybe 4 or 5 trials. So, but yes, so there's certain predictions that arise from what Josh is bringing up. So at some point, how far back can the memory go? And this is a very important question in relation to the time constants of the hidden states. So yes, it can only go back to certain past events so far back in time. But again, that's what we do. And think about it this way. If you slow down Morse code or speech so much that the speech note is a second or 2 seconds apart, it's no longer Morse code or music or speech, right? You can't understand it. The argument we would make is that timing in this scale is very special because it's only at this scale that you have these hidden states, that you can tell time or accumulate information about past events in a context of any time. So, so a prediction that arises is that the intrinsic timing hypothesis argues that timing is such an important computation, virtually all networks are inherently capable of some forms of chemical processing. So the prediction is that time can be observed or even learned in vivo circuits. Now, so I'm going to show you some experimental data now. But I want to highlight something. So we're going to try to teach circuits in vitro to tell time. And this is an important approach in our labs, an important part of our philosophy, which is that, as you'll see in some of my future slides, even when we simulate a cortical network in our computer, it doesn't take much to make it so complicated that we don't understand it. There's guys at Google that are developing these incredibly sophisticated deep neural networks that we use for discriminate Siri, recognize for speech recognition. And the truth is they don't understand those networks. They don't quite understand how they work. They know they recognize faces or speech, but they're struggling to understand how they work. Now, there you have a system in which you have every single variable available. You know each synapse, you know each rate each neuron does. Yet they don't understand it. It doesn't bode well for us, right? Because here we're stuck with a couple of billion neurons in the cortex without access to that information. So how are we going to understand that if it's already hard to understand that in a single system? So one way we believe it's important to make this more tractable is that if we can coax in vitro circuits to act as a chip, as a computational chip, in which we can teach those circuits to do some important computation or network level computation that's interesting. And this is but we have to teach them something, so that's the first thing. And this is what Anu Goel in my lab has Anu Goel in Carlos's lab has been working on. Anu recently joined Carlos's lab. So we know that even in vitro, cortical tissue can, in a sense, has dynamics and tell time. So this is a recording from an organotypic slice, just recording from one pyramidal neuron in response to electrical stimulation. So this neuron has the monosynaptic EPSP, but there's also network dynamics. That's internal, recurrently generated activity that causes this continuous firing. And this neuron happens to fire around 200 milliseconds. So in a sense, it contains information about elapsed time. And this is over multiple trials, over 20 minutes. It fires with some degree of reliability at 200 milliseconds. But here, this is just luck. We're not able to control the system. So what Anu did is she asked, can we teach the circuit to tell time? So she used organotypic slices transfected with channel rhodopsin and used a protocol which is a bit similar to a classical conditioning protocol, in which a CS, electrical shock, is paired with light. So you have shock light at different intervals, and you have a control pathway, CS minus, which is unpaired or the non-paired pathway. So this can be done for 2 or 4 hours in the incubator, just shock light, and then test. Now you're just testing in response to the shock. So here's her protocol. Some slices were trained at 100 milliseconds, 250 milliseconds, or 500 milliseconds, pairing at different intervals, and then testing. The test is the same, you just activate your CS plus pathway. So here's a recording from a single neuron in each one of those groups. Here's your monosynaptic EPSP directly activated by the axons that you're stimulating. But here's the internal dynamics of the network. The second polysynaptic EPSP happens early here. It's not very, the internal dynamics is short lasting. Here it's a bit longer lasting, and here it's even longer lasting. So the question is does this tissue care about its experience? Did it learn? Did it extract anything from what it experienced for 2 or 4 hours? And the answer is yes. That world of that slice is our external stimulation, and it matters to the circuit. So the internal dynamics of the circuit is more short-lasting in this group compared to the other group. So I'm sorry. So here is now all our data from all our cells, all our trials. This is the voltage normalized to the peak and sorted by the time of the second peak. So here you can see that the dynamics is relatively short-lasting. Here it's medium-lasting, and here it's a bit longer-lasting. So the experience of the tissue has altered its dynamics. In a way, that reflects time. So here's average data now across all the trials in which there was polysynaptic activity or dynamics, just average, raw averages across all the activity. And the ones trained for 500 milliseconds, there's more activity later on, and a bit over here at 250, and a bit here at 100. So there's, even at the average level, which is not the best way to look at this, there's differences here. But if you look at the timing of when the cumulative distribution of the events of those polysynaptic EPSPs, there's significant differences in the distribution, as well as in the median polysynaptic event time, when those polysynaptic peaks is occurring on average. So they occur on average later if that slice experienced a longer interval. So one interpretation here, and we have to be a bit careful with interpretation, is what the network's learning to do is what we do in a sense, which is create an expectation of what should be happening. If the light comes in at 100 milliseconds, or 250 milliseconds, or 500 milliseconds, can the circuit adapt to create an expectation of when the next pulse of activity should come in? And that's what we're trying to get to. Now, how does this happen? We don't know the cellular synaptic mechanisms. We're still looking at this as a network mechanism. But what Anu did, for example, one hypothesis is that what's happening is the trained pathway, the trained pattern of activity is more burnt in or consolidated or consistent, or in terms of correlations between any two cells in those networks. So if you have, now she's recording from 2 neurons simultaneously, and she's looking at the level of correlation between those 2 neurons. And in response to the paired pathway, the correlations look pretty good. In the unpaired pathway, the correlations might not look as well, but then that is borne out here by the correlations between, in the same cells, in the same slices, in response to different pathways, is higher in the paired pathways, meaning that the neurons, that trajectory, or at least the neurons, are more tightly coupled to each other when they received paired shock and light. Now, there's a couple of controls you have to do here to determine what's the cause of that, but if anybody has any questions, you can ask me later. Now, the other thing she asks is, well, what about the balance of excitation and inhibition? So there's more activity during the time pairing. So using traditional methods here to look at the balance of excitation and inhibition by clamping the cell at the reversal potential for excitation or inhibition, she calculated the balance of excitation and inhibition in response to the paired pathway or the unpaired pathway, the non-paired pathway, at different time limits. Right in the middle, right during the pairing where the light was occurring, or after or before. And what she saw is that the balance itself is dynamic. There's an increase in the EI ratio right at the paired time. So this highlights the challenge, because it's not that you can simply say, okay, there's more excitation, more inhibition. The balance is dynamic. There's more excitation at that point in time, but at the other points in time, there's no significant difference between the paired and not paired pathway. So this, again, brings to mind the challenge in looking at the system as a static entity. So we think that what's happening here is that you have dynamics, recurrent dynamics generated by the internal dynamics of the system, which is very challenging to understand. To approach that, I'm now going to switch back to some of our computational work, but that's even a bit simpler than the computational work represented at the beginning. So this is because it's going to use firing rate neurons, and I'll tell you what this is. So here, to attempt to get a grip on the dynamics of these networks, we're going to use computational methods. And we're taking this, or extending work that was very classical work by a neurocomputational scientist called Heinz Sempowinski, done way back in the 80s, and more recent work by Herbert Jaeger and Marietta and David Cecil. And what they do here is we have a network, a simulated network, in which we have these units. And these units are just sigmoidal sort of analog units that go from a certain value, like -1 to 1. And then we call those firing rate units, and they're in a randomly connected recurrent network. And this equation here tells you, the neuroscientist, exactly what you know it already is. All this equation is saying is that the voltage or the activity in this cell is a function of the voltage in the previous time step, the input from its neighbors, times the weights, times any external input by the black, plus any noise in the system. So this equation is just capturing your intuition that what this neuron does is determined by what its neighbors are doing times its weight. Now, one thing that Sempowinski showed is that these networks, when you randomly connect them together, they have two regimes. They can be what we call a low-gain regime or a high-gain regime. Both of these are fairly intuitive. If you imagine everybody here is a neuron, and people in the room don't like each other, they're far apart from each other, and they don't have strong coefficients or weights between them. So if we activate some of the neurons, maybe we push some people in the front row, that activity won't be propagated down the street. So it dies out, it fades away, because the recurrent connections aren't strong enough to transmit information. Now, in the high-gain regime, when the coupling coefficients between everybody, the weights are very strong. So when we activate the network, it creates these perpetually changing patterns of activity that go on and on and on. Computational neuroscientists know that these regimes are much more relevant in terms of their ability to process information and to store information. The problem is, and the problem for decades, has been that those are the regimes that are chaotic. So what does that mean? So here, now this is a simulation of 100 units out of 1,000. And the color represents if it's on or off or low or high. And it's just, this is the trajectory of the system, the pattern at this point, the pattern at this point, the pattern at this point. And that's the input to the system. Here I'm showing you the voltage trace, if you will, of 3 neurons, 1, 2, 3, on 2 trials, one shown in blue and one shown in red. And you see that when we give an input across trials, these 2 traces overlap a bit, the blue and the red, because you have started off the system in a similar part of state space, meaning the activity pattern of all neurons. But you see that the blue and the red rapidly diverge. So you quickly lose information about what happened in the past. By the time you're looking over here, you can't tell when this stimulus occurred. That's the definition of chaos. Chaos means that very small changes at one point in time produce drastic changes at later points in time. So very small differences over here end up decorrelating all the activity over here. So chaos voids the ability of this network to tell time, to store information about what happened in the past. So what we wanted to know is if we can sort of preserve the complexity of the patterns, but tame the chaos by tuning the current weights. This is a very natural thing for neuroscientists, right? We know the weights, the synapses are plastic. So what we did is ask, well, if we now make these weights plastic in our network, and train the network to produce a pattern, what we call the innate pattern. So you train the network to reproduce any pattern, such as this pattern here, over and over and over again. So each unit you're training to reproduce the pattern that it already produced. It's a reference pattern, it's an innate pattern. And you can adjust the weights to make that happen. And if anybody has any questions, we can do that. It's a highly supervised move. It's not really biologically plausible at this point, and that's one thing we're struggling with. But once you do it, you go from this chaotic activity to this highly reproducible pattern activity. So now you give the input, and it will play the same thing over and over and over again, at least for certain periods of time. So if this pattern in the current network is being played over and over and over again in response to the same input, that means you can now use the output to do something useful, like tell time, like maybe generate a pulse at 2 seconds. So, in other words, think of this as a short-term memory. For this to fire here at 2 seconds, it has to remember that there was an input 2 seconds ago, right? So this, we look at this as timing. Other computational neuroscientists look at this as memory. The memory of this relates to Josh's question. If you keep on giving about many times, how far back in the memory of the system you go before it converges to a steady state? So once you've done this, now, and this may not be obvious, but it's a fact that if this is a high-dimensional trajectory, you can now use this to generate many different relevant outputs. So now we're going to train these units, x and y, to be the pen on a piece of paper, two-dimensional position on the x and y axes. So now we give an input. This generates a pattern, and we could use that pattern to just drive another pattern in two-dimensional space. We have a pattern in 1,000-dimensional space. It's relatively easy to generate any pattern you want in two-dimensional space. And that two-dimensional pattern is the word neuron. And a property that's relevant here is that these patterns are now themselves attractors. Neuroscientists have often thought of the brain as operating as fixed point attractors, meaning the memory is the activity converges to a steady state. But what we're trying to express here is that the memory, or the computation, is precisely the voyage through state space. It's the pattern as it is occurring. So here, if we give it input 1, that triggers the pattern that we've trained these guys to spell chaos. And then we perturb the system right here. We inject a lot of noise right here. It will screw up the output. And that's what you're happening right here. But the network has a memory of what it was doing. It can come back. This is an attractor, so this is the opposite of chaos, right? Normally, you'd have the perturbation and it would diverge exponentially. Now it says, okay, I was doing this, and it comes back to complete that pattern. And it can do that here or here. So now the notion is that you're encoding information in the dynamics of the system, in what we call the dynamic attractor. Now, that's at the motor side. What Vishwa Godar in my lab wanted to know was, can the same network process both sensory time and motor time? So what he did was look at a challenging task to ask if the same network can both discriminate speech sounds and transcribe them into a motor pattern. So here he gets a spectrogram of spoken digits, converts that into an analog input of very few channels, 12 inputs, and feeds that into a recurrent neural network that was trained in the manner that I told you about. And now there's 3 outputs. The X, the Y, and the Z would be the pen, position of the pen on the piece of paper. And so he trains this to learn this pattern, the pattern produced by this, this input. And then he trains these guys to produce this. So this is the training. This is what the testing looks like. So now he gives it different utterances of the word 0, 0, 0, 0, 0, all of the same subject, by the way. And this is the output. So the network receives this input and generates this output. So the network itself is transcribing spoken speech, so doing both a sensory task and a motor task, both of which contain temporal information, sequences, patterns. And shown in red here is patterns that were trained, and green were naive patterns. So this is an example of how the same network can do both sensory and motor tasks if you're encoding information in the dynamics of the system. So to wrap up, I'm just going to give you a demo here, because I realize that sometimes it's better to get intuition by seeing something work. So here, in this simulation, we have a simulated network of maybe 1,000 units of these firing rate units. And I'm just showing you the activity of 10 of those units, and I'll slow this down a bit. And so this unit happens to go on, then off, up, down. It's just changing. There's no periodicity. There's no obvious limit cycle here. It's just changing in high-dimensional space. This network has already been trained to be stable in response to input 1 and input 2. And these outputs have already been trained to produce the words chaos and neuron. So when we give the appropriate input, this now generates a pattern, which is transformed into another pattern over here. If I give it this pattern, it generates another pattern in high-dimensional space, which is transformed into this low-dimensional pattern. And the important part here is once we do this, if we perturb it right there, we screwed up the O, it returns to that pattern. So it has a memory of what was stored in the system. Okay, so to conclude, state-dependent network model proposes that temporal processing is a general and intrinsic computation of cortical networks, one that emerges from the natural dynamics of recurrent networks and the universal presence of time-varying neural properties. Evidence that even cortical networks in vitro seem to be able to learn, if you will, temporal structure strongly supports our hypothesis that time is an intrinsic computation of neural networks. And finally, recurrent networks are very powerful computational systems, but it has been challenged to exploit this potential in part because of chaos. We now know, at least in principle, that by that chaos can be tamed in recurrent networks, resulting in dynamic trackers, which store spatial temporal objects in memory. The problem is, we still do not know how to tame the chaotic dynamics in an unsupervised fashion or in spiking networks. So these are things that the field, our lab and the field, is struggling with. So lastly, let me thank my lab, everybody in the lab, but I focus more on Buonomano's lab as I work. But I have a said she's now in Carlos's lab, so I might have to remove her from that point. And Vishwa Godar, and also the Neurobiology work. So thank you very much. Jack.
So there are lots of other rhythms in the brain, gamma rhythms, alpha rhythms, and whatnot. Are these rhythms things that these neurons learn to do and are part of the training, or they also have the ability in the network to do some kind of processing just because they're right on top of the oscillation?
I'm surprised you're asking about oscillations. I don't know the answer to that question. I mean, as you know, better than anyone, in the cortex, the computational role, function of things like gamma and theta is debated. These models can certainly exhibit those oscillations, but their function is not clear. One possibility, again, as I'm sure you know, is that it's just synchronizing the output so the next stage of the system has increasing the signal-to-noise ratio. If it's just doing that or something more profound, is not clear. The irony here is that people have used those oscillations as an argument for it's using those oscillations to tell time, meaning there's another system that's counting those. We argue against that. Yes, you have oscillators throughout the brain, but to my knowledge, there's very limited evidence that there's any other circuit that's counting those oscillations. Any other questions? Mark?
Totally fascinating. There's sort of two related questions. And one is that, owing to the fact that it's intrinsic dynamics that are, there must be some constraints that are therefore set. You can't arbitrarily shift into perceptually short time or very, very long. We couldn't get arbitrarily faster at what we do and how we perceive. But that there is there's some flexibility. You know, other animals, small animals, insects, they fly around at high speed, they perceive sensory reality on a very different time scale than we do. But they're, you know, the dynamics of the neural circuitry that do this don't seem radically different, aren't operating radically faster, for example.
Yeah, I mean, I think it's a question of perception and processing, right? So let's just say processing at very high rates. So I think that what you refer to as a bandwidth is there's a range in which we can do certain things. So maybe tens, hundreds of milliseconds or a few seconds, we can control our movements. Outside that range, that falls apart. So the prediction is that the properties of, say, doing computations in which you can interpret Morse code, for example, in the context of what came before, only happens within the range. But that range corresponds to much of what we know about the time constants relevant for synaptic dynamics and channel kinetics, right? Tens, hundreds of milliseconds. So we would argue that it's not a coincidence that speech and music have the same time constants as the physiological properties of the motor neuron. And so that's limited to that sort of. Mark, you can bring this up if you want.
Any other questions? No, thank you very much, Steve.